Hello, Assalamu alaikum wa rahmatullahi ta'ala wa barakatuhu. Ladies and gentlemen, my name is Nuruddin Sani. I'm here to present on the biology content for SS2, first term, week 1. Our topic of today is classification of plants. Plant classification simply means grouping of plants according to their similarities. So plants show kind of similarities in terms of their morphological structures, like leaves, like flower, pattern of the flower, shape of the leaves, types of the roots. These are all morphological characters by which plants can be classified. The next slide talking about the basis on which plants are classified. Plants are classified using the following criteria. 1. Based on physical characteristics. Classification based on physical characteristics, that is what we often refer to as botanical classification. So plant can be classified as flowering and non-flowering plants. That means plant bearing flowers are referred to as flowering plants, while those without flowers, we call them non-flowering plants. We can also classify plants based on the life cycle, as in annuals plants, biannuals, and perennials. When we say annual plants are those that carry out their life cycle starting from vegetative growth passing through flowering or reproductive growths in 1 year, while biannual plants are those plants that can finish their life cycle within 2 growing seasons. That means vegetative growths and flowering growths or reproductive growths within 2 growing seasons. That means 2 years. While perennials are plants that can do their life cycle, or that can start their life cycle up to the complement of the life cycle within more than 2 years. We can also classify plants based on uses as either food crops or non-food crops. Plant kingdom is divided, looking at it on the basis of possession of flower or on the flower character. So plant can be viewed as flowering plants and non-flowering plants. The non-flowering plants are subdivided into 3 groups: the talophytes, the bryophytes, the pteridophytes. So we'll take this grouping or subgroups under flowering plants one after the other and explain them in details. So the talophytes. These are the simplest organisms which had cell wall. So possession of the cell wall make them to be plants, or make biologists or botanists to classify them as plants. They lack structures like roots, stem, and leaves. They include bacteria, fungi, and algae, as we can see in the diagrams below. The most renowned among these are mushroom and nitrogen-fixing bacteria. Many are notorious plant pathogens causing serious diseases of plants like rust and smots. So this it is a typical diagram representing one of the members of the fungi, often referred to as mushroom. As you can see, the parts of these organisms were labeled. This is scales, this is carps, rings, gills, which the organism often use to carry out gaseous exchange. This is what we often refer to as stem, the cuff or vulva. So the whole of this organism is a member of fungi and is termed or is named mushroom, and it often grows in area where there is a lot of organic matter, and it accelerates the decomposition of organic matter, thereby increasing the humus content of the soil. The next one here is a typical bacteria. These capsules, pili, cell wall, cell membrane, nucleoid, ribosomes, cytoplasm, and flagellum. And these are all part of the cell organelle, part of the cell which you often refer to as cell organelles that carry out functions or that enable the bacteria to carry out its physiological processes. For example, like this flagellum is one of the organelle that enable the organism to move from one place to another. If you look at the diagram, C is representing algae. These are here are groups. This is cyanobacteria, green algae, and diatom. So this is a group of different members representing algae. So all these examples, the bacteria, mushroom, and algae, they all belong to the subgroup Thallophytes. They possess cell wall, but they lack true root, true stem, and leaves, and that is why they were categorized or they were classified as such Thallophytes. Then the bryophytes, the next group. These are mosses. Examples include mosses, liverworts, and hornwort, which lack true roots and flowers. They lack true roots and flowers. Their representative members are presented here below, or they are being drawn here below. You can see this is the moss, this is the liverwort, this is the hornwort. These species are ecologically very important in the trophic and in the forest ecosystem. The next group is Pteridophyta. These are higher plants having vascular system. So unlike the Thallophytes, the bryophyte, the Pteridophyte are more advanced because they have vascular system. I mean the system that conducting system. That means the xylem and the phloem that ensure conduction of water and translocation of manufactured food from the leaves of this plant down to the root for storage and to enable the plant carry out their physiological processes. They also have true leaves, stem, and roots, which is another advancement over the Thallophyte and bryophytes. They do not have flowers and no fruit formation. This is one of their weak points. Even though they are considered to be the higher plant, but yet they do not bear flowers, as the most advanced group of plants show flower formation as well as fruit formation. So hence reproduce through spores. Since they do not have flowers, they bear spore formation. So whenever they release their spores, the spores will be carried by air or any other agent on landing on suitable environment or favorable environment. They germinate into new plants. For example, pans, club mosses, hostel are examples of members of Pteridophyta. Are commonly found in trophics and forest ecosystem, and they have agronomic use as ornamental only. So the higher plant, the flowering plant, as we can see there, the plants were divided into non-flowering and flowering. So we have seen that the flowering plants, the non-flowering plant, were subdivided into three, as shown in the previous slides. We have the Thallophyte, we have the bryophyte, we have the Pteridophytes. So now we enter into another group or subgroup called flowering plant. These flowering plants are further divided into two groups. That means monocot or monocotyledon and dicotyledoniae. Monocotyledoniae and dicotyledoniae. This group of plants, they produce flowers. Another way, we also call them angiospermae or angiosperm. Monocots, the subgroup of angiospermae or flowering plant. They have single cotyledon in their seeds. What it means by single cotyledon in their seed, meaning that they have one seed leaf. That means when we plant them, they germinate, one leaf will appear from the germinating seeds. Their germination is hypogeal. Hypogeal simply means that the hypocotyl remain below the surface of the ground. Most important cereals and fodder belong to these groups. They have parallel venation on their leaves. That means when you take their leaves, looking at the surface of the leaf, you see the pattern of the veins, they go parallelly from one polarity to the end. They include wheat, rice, maize, barley, sorghum belonging to this family called Poaceae. So these monocot plants, one of their characteristics, the moment you take their leaves, you look at the lines pattern in the leaves, they go parallelly. That means from end to end. So wheat, all grasses, rice, maize, millet, barley, sorghum are example of monocots. Looking at these monocots to further visualize their characteristics, one cotyledon in the seed, leaves with parallel venation, vascular bundles scattered throughout the stem. As we said, the flowering plant, they have vascular bundle or vascular tissues or vasculature. When we say vasculature, means conducting tissues. And these conducting tissues are xylem, which is a structure in flowering plant that ensure transportation of water from the roots up to the leaves for photosynthesis. And then the phloem, which is a tissues that conduct manufactured food from the leaves of the plant down to the roots for storage and to other parts of the plant for their physiological processes. So these monocots, if you look at the anatomy of their leaves, stem anatomy or root anatomy, you will see that these vascular bundles, that means the xylem and the phloem, they are scattered in their anatomy. So root system composed of many fibrous root with many hairs. So if you look at their root system, they are radiating from the same axis, they are of the same length. This type of root system is what we often refer to as fibrous root system. You can see it in onion, you can see such type of root in maize plant, you can see such type of root system in wheat, rice, barley, sorghum. They have similar pattern. So the moment, even without uprooting the plant, the moment you study the leaves of a particular plant, and it happens that the leaves has parallel arrangement, parallel venation, that means the line of the veins go parallelly. So there is no doubt about it. Even without uprooting the plant, you know such plant must have fibrous root system. And if you want to practically observe it, uproot the plant, you will bear me witness that the root system will look fibrous. They all radiated or originated from the same axis, and virtually they will be of a similar height. Flower part generally in three. That means part of the flower, the floral part of the flower are sepals, petals, stamen, and carpel. So most of this external wall, that means the sepals and petals, they are always in threes, either 3, 6, something like that. Multiple of threes, they appear like that. Not even, they are in threes, they are in odd. Then the other group is dicot. They have two cotyledons in their leaves, in their seeds. That means there are two seed leaf. There are two seed leaf, meaning that when you plant them, they germinate. The two leaves will sprout from the germinating seed, just like what is obtainable in beans, mango. When you all plant them, you see two leaves will emerge. They are the most advanced plants. Their germination is apigeal, meaning that the epicotyl appear on the surface of the ground. The plants are broad leaf and have network venation, unlike the monocot that have parallel venation. But this one, their vein system on the leaves, they are networking. There is interconnectedness. They appear like just like a network system of veins. So many of them are economically important to man for either food, fiber, or other purposes. So they include soybean, pulses, tobacco, cotton, groundnuts, mango, and rest of them. So as from today, for you to ascertain whether a plant is monocot or dicot, right from the leaves of that plant you can be able to conclude whether a plant or a given sample of plant is monocot or dicot by just looking at the leaves arrangement of veins rules arrangement of veins on the surface of the leaves. So if the arrangement of the veins on the surface of the leaves appear to be parallel, meaning that these plants or those group of plants you are observing, they are what? They are monocot. And if the arrangement is networking or network or reticulates, meaning that they are dicot. Below here are other characters of this dicot. Is just a summary. Two cotyledon in the seed, leaves with network veins, vascular bundles for palm rings outside the stem. So looking at the anatomy of the stem of this dicot, you will see that the xylem and phloem, they arrange in such a pattern, they form ring pattern instead of scattering like what is obtainable in monocot. Root system is typically composed of primary tap roots and many hairs. So in this type of arrangement in dicot, the root system is appear to be there is one major root called tap root. Onto that one major root, other roots radiate, which we call lateral roots. So flower fat generally in fours or in pipes, meaning that flowers are either 4 sepals or 6 petals or pipes times 2 making 10. They are always even, not odd, as what is obtainable in mono. So here we are trying to make comparison between monocots and dicots. Monocots, the leaves in they have one seed leaf, so when you grow them or when you plant them they germinate, only one seed, only one leaf will emerge from the seed. While the dicots, they have 2 cotyledons inside them. That is why when you cook beans, the seed or the bean seed can be split into 2 equal parts, indicating that it has 2 cotyledons. Granite, when you split it, when you cook granite and you split it, it will split into 2 halves, indicating that it had 2 cotyledons. But coming back to plants like what? Maize. No matter how you cook maize, you cannot be able to split it into 2 equal halves. Similar to rice, similar to sorghum, similar to millets. So such plants or grains or cereals which you cannot split their seed into 2 equal parts after cooking, it is an indication that they contain only 1 cotyledon, that means 1 seed leaf. Another difference, another criteria to differentiate between monocot and dicot is on the basis of veins arrangement. Looking at the arrangement of the veins, they are now parallelly here. They go from one axis to the end, one axis to the end. This is what we often refer to as parallel venation, leaf venation. Leaf venation here, instead of being it parallel, here is network or net-like. You see they are spread just like network instead of going from one end to another parallelly. On the basis of stem anatomy, looking at the monocot, the vascular bundles, see the vascular bundles here, they are scattered along the stem. They are scattered. So looking at the vascular bundles here in the dicot, you see they form arrangement pattern. They arrange themselves in such a way that they form a ring structure. So this structure that connects the vasculature is what we often refer to as cambium. Then another character by which we discriminate or we differentiate between monocot and dicot is by looking at their roots. This is what is called fibrous root. You see the roots are originating from the central axis, from the same origin. This type of root arrangement, we call it fibrous root system. While in dicot, it seems to be like there is one mother root. Look at it here, one mother root starting from these points, going down. But smaller roots are attached to the mother roots. And these smaller roots, as you can see, is what we call lateral roots. Then coming to the flower, which is also another character by which we can differentiate between monocot and dicot. So if you look at the sepals here, let us count the sepals here. 1, 2, 3, 4, 5, 6. So plural part usually are a multiple of 3. Multiple of 3, that means 6. 2 times 3, making what? Making 6. They are a multiple of 3. But looking at here in dicots, count the number of petals and see. 1, 2, 3, 4, 5. Sepals 1, 2, 3, 4, 5. So plural part usually are a multiple of 4 or 5. So multiple of 4 or 5. So they may be 10, they may be 8, something like that. Thank you for listening.
You can subscribe to our YouTube channel, Exponential. You can subscribe to our YouTube channel. You can subscribe to our YouTube